Good evening to one and all present here. Myself, Ajit Kumar, a PhD scholar from Department of Bioscience and Bioengineering from IIT Guwahati. Today we will be seeing week 6 live interaction session on the course Environmental Biotechnology with course ID NOC 25BT83. BT83. I hope my voice and PPT is visible. And now we will start this session. And this session will be comprising of the week 6 recap of environmental biotechnology. We will see first what are all the important concepts that has been covered earlier, and that will be followed by the MCQs, sample MCQs of week 5 and week 6. And I hope as everyone completed their assignments of week 6, and we will move forward for the recap of environmental biotechnology course. I hope my voice and PPT is visible. Okay, then we will start the session. Then we will start the session. Before moving on to the session, I will introduce myself. Myself, Ajit Kumar, joint degree research scholar between IIT Guwahati and IIT BHU Varanasi. I work in the laboratory of biochemical and environmental engineering, where our lab works mainly on the wastewater treatment, where my research inclines on the application of enzyme in wastewater treatment, especially textile wastewater treatment. Moving on to my qualification, I have done BTech Biotechnology from Shastra University and MTech Industrial Biotechnology from NITK Karnataka. And about my work experience, I worked in Biocon and Enzyme Biosciences, biotech pharma companies. And this is all about my personal introduction. We will move on to the week 6 and week 5 recap of what are all the important concepts that has been covered so far. And before moving on to that, in the last class, in the last NPTEL week 5 live interaction session, we have covered growth of individual cells and their proliferation and differentiation concepts. Then we have seen detail on the cell cycle, where we have seen different phases of cell cycle, which is G0, G1 phase, S, G2 phase, followed by M phase, which is mitosis phase. We have seen all these phases of cell cycle, and also we have seen the concept of exotrophy and endotrophy in the phases of cell cycle. And moreover, we have seen the growth curve and different phases of the growth curve, where we have discussed in detail about the lag phase, followed by the log phase, followed by stationary, and finally the death phase. And regarding to that microbial growth, we have seen many equations, how the number of cells can be determined, and the growth efficiency, YXS, YPS, we have seen, and many more equations in order to find the microbial growth in an ecosystem. Then finally, we have seen the concepts of batch and continuous culture system, and we have covered different bioreactors, which is aerobic and anaerobic configuration of bioreactors for both batch and continuous system. So these are all the topics that have been covered in the last class, and today we will be seeing some more important concepts. And the first concept is that we will cover is the physiology of the microbial environment, where first we will see how selective pressure causes the physiological diversity of microorganism. In order to understand that, let's think why these microorganisms are incredibly diverse. If you think on the answer will lies in due to their diverse habitat, they This diverse habitat, different environmental condition makes the microorganism undergo some kind of pressure over the evolutionary time. So what happens here? The microbial microbes they adapt to the specific environment that they live in. So by adapting to the specific environment, their metabolism changes drastically over the evolutionary time. And if we see the three terms that defines the three terms that defines the life is metabolism, replication, and hereditary. And if you see the each term explanation and the definition, the metabolism is what? It is nothing but the way they obtain energy. Either they obtain energy through catabolism or they are using the energy in order to make new building, which is anabolism. The concept of catabolism and anabolism we will be discussing in the upcoming slides, but metabolism is one of the key factor. The metabolism is one of the key factor that governs the life form. And the second concept is that replication. What do you mean by replication? It is nothing but the ability to make copies of themselves. As we see in the DNA replication, right? The DNA is replicating in the exponential manner. So this is what we call as replication, where the ability to make copies of themselves.
हेलो सर।
Hello?
सर इसमें आरएनए का भी कोई फैक्टर होता है?
Like what? RNA?
रेप्लिकेशन में सर,
Ah,
रेप्लिकेशन।
yes. Yes, it includes RNA also. To give an example of that, I have given DNA replication. So
थैंक
that
यू सर।
brought, yes. Thank you. Thank you, Mohit. It can be RNA or DNA or anything. Replication in general means the ability to make copies of themselves, right? It can be applied to cells also. Cells also replicates, right? One cell is replicating into two, two replicates into four in a binary fission. So in order to give one example, I have given DNA. So it applies to a general form. And the third term is hereditary. Hereditary means passing some information, passing the trait from one generation to the another generation. So these three, metabolism, replication, and hereditary, are three important critical traits of life. And what happens? They don't happen in isolation. So what happens here? They depend upon the environment, and therefore the diversity. This physiological diversity they result from the billions of years through environmental pressure that shaping the metabolism, replication, and hereditary. So for all the life form, they have metabolism, replication, and hereditary function. And this is how they undergo selective pressure over the evolutionary time. Then, as we have seen in metabolism, the catabolism and anabolism concept, you need some kind of energy or source in order to process this metabolism. So the important two energy sources are the carbon and energy sources. So what happens here? The whole form of lives, starting from prokaryotes to the humans, they need some kind of sources to make the living. In this case, it can be carbon, energy, or electron sources. To achieve this, we require at least two physiological sources. One is the energy source. Why it is needed? In order to generate ATP. And we all know that ATP is energy currency of the cell. Without ATP, what happens? The life doesn't exist. Our body doesn't get the appropriate energy to function themselves. And second important factor is their carbon source. Why we need carbon source? To assemble the cellular building block during the maintenance of the existing cells or to produce new cells. So these two are the main important factors that are needed for their survival. In addition to that, electron sources are also needed. but at least they need two physiological resources. One is energy source in form of any energy. It can be NADH, NADPH, or ATP. Ultimately, these will be converting into ATP for survival or for metabolism of their own cells. And second is their carbon source. What for? For their maintenance, if it is an existing cells, or in order to produce new cells, we need carbon source. The carbon source can be anything. It can be a simple sugar source, which is glucose, and we will be seeing in the upcoming slides the different sources. And if we see the flow chart of the catabolism and anabolism concept, as we will be seeing more in the upcoming slides, but to a simple term, the catabolism is nothing but breaking down a oxidizable substrate, for example, sugar, for gaining the ATP, energy molecule. So, catabolism is nothing but breaking down of the substrate for gaining the energy. Energy means it is ATP. Whereas anabolism refers to using the ATP or the energy that are generated from the catabolism to form the macromolecule or new cells, or to give maintenance support for the already existing cells. So these two concepts we will be seeing how the carbon and energy sources are used. And as we know that energy source can be chemical or light. So if it is used as the chemical energy, a chemical as an energy source, we call it as chemotrophs. If it is uses light as an energy source, we call it as phototrophs. And the carbon source, it can be any source. For example, in this example, we have taken the sugar. What happens here? First, the sugar will be transported into the cells via different transporter molecules, and they catabolize. Catabolize means they will break down this sugar molecules into simpler compounds. First, they will undergo glycolysis cycle, and what happens? The sugar will be converted into pyruvate, and pyruvate is the precursor molecule. And if it is undergone a fermentative metabolism, then it will be converting into some other metabolite like lactic acid or ethanol or some kind of other metabolic products. If it is not going fermentative metabolism, instead it is undergoing TCA cycle, what happens? It will be converting into acetate, acetate molecule, and that will be undergoing oxidative phosphorylation where oxygen will be used as an electron acceptor, and it will be eventually forming an H2. If you observe this cycle either in the glycolysis or TCA or oxidative phosphorylation, you will observe the ATP molecule will be formed at each stage, and also other reducing equivalent like NADH, NADPH, or FADH is also formed, and these are all the molecules that provides the energy to the cells. So this is the catabolism reaction where what happens, as the definition suggests, the carbon source sugar is breaking down and they generate appropriate amount of energy in the form of NADH, NADPH, or ATP. So this is how the sugar is converting into energy form. And if you see here in the anabolism process, the already generated ATP will help these precursor metabolites like simple monomers or amino acid in order to biosynthesize them and form building block. And how do they form? With the help of the ATP that are generated from the catabolism process. And these building blocks will be, these monomers will be polymerized into macromolecules like proteins or DNAs or RNAs, any kind of substance that are used for their own metabolism. Here it can produce the carbohydrates, it can produce a fat, it can produce proteins. So this is how the with the generated ATP we are generating back the organic compounds, that is macromolecules. So by this flow chart we understand that the catabolism refers to the breaking down for their energy, whereas anabolism, they are building up the compounds from the energy that is spent during the catabolism. So, in one, the energy is available for cells to build the new things. So, this is how the importance of carbon source and energy source in catabolism and anabolism exists. So, after understanding this metabolism process, what environmental biotechnology needs to understand is the first one, what drives this metabolism and what are all the energies and carbon source that are seen in the particular environment. So if you want to understand the microbial ecosystem, as we have seen in the earlier lectures, the first question that we need to ask is what drives this metabolism here? What drives the this process, either aerobic or anaerobic process? What drives the system? And next question is, what are all the energy and carbon sources that are available for the microbial system in order to drive this metabolism? And if you could answer these two questions, then our understanding of microbial physiology and microbial ecology is up to the mark. So the answer is provide a nutritional basis for carrying out this biochemical and ecological process. So, if you see again the type of organism, as we have seen, based on the electron and based on the energy and based on the carbon source, there are different types of organism that prevail in the environment. And if you see the first type of organism, which is chemi-synthetic organotroph. And what does it mean? Chemi-synthetic organotrophs. It is nothing but it will use organic compound as both energy source and the carbon source. Right. Chemisynthetic organotroph is nothing but it uses organic compound as both energy and the carbon source. And if you see the chemisynthetic lithoheterotrophs, what happens here? It uses inorganic compound as the energy source and organic compound as the carbon source. This is how different microorganisms are classified based on their carbon, energy, and electron source. And the third one is photosynthetic heterotrophs. What happens here? The term itself clearly defines that it uses light as an energy source and organic compound as the carbon source. Whereas in chemisynthetic lithoautotrophs, it uses inorganic carbon or inorganic compound as the energy source and also inorganic compound as the electron source. And if you see the carbon source, it uses carbon dioxide as their carbon source.
Sir,
So if you see the chemisynthetic lithotrophs, chemisynthetic lithoautotrophs, what happens here? It will use inorganic compound as an energy source and inorganic compound as the electron source and CO2 as carbon source. If you see photosynthetic autotrophs, what happens here? It will use light as an energy source and CO2 as the carbon source. So uh, the this is how the microorganisms are classified based on their energy and carbon source. And if you see, and if you see the, as we have seen, the selective pressure, what does it mean? Selective pressure is nothing that due to some adverse condition, it can be an higher temperature or it can be an acidic environment. It can be any kind of adverse condition. So how this nutrient flux is happening in the ecosystem and uh, and how it and this adverse condition, how it's shaping the physiological status and composition of microbial community. So this is one of the important concepts that you need to know. Again, it tells us how the nutrition flux, that is how the transfer of carbon and energy sources helps in maintaining the physiological status and composition of microbial community. The first that you need to understand is that in any environment, you need to understand what are all the resources that are available in the environment. And next is what is the nutritional status of that particular environment and geochemical composition of our biosphere. These three important aspects help us to understand the pressure for genetic selection and adaptation. And before moving on to the core concept, we have to ask these questions in order to understand ourselves. And first, what was it like to be an early microorganism in the Earth predominant seas? And what kind of pressure, either it is a physiological or ecological pressure, that the microorganism faced in the either an aquatic or terrestrial habitat? And in the particular habitat, what was their carbon and energy source that are available for them to drive the metabolism? And how important is their nutrient uptake? Whether these microorganisms are short or between the microorganism there is any competition or predation or parasitism are observed. So you have to understand these kind of observation in the ecosystem in order to understand their behavior in a particular environment. And to which degree do issues, this tells us that how much extent the microorganism can sustain, whether it can sustain the acidic stress or oxidative stress or temperature extremes or UV irradiation influences the physiology. So these are all the some of the key questions that needs to be answered before moving on to understand the metabolism. And the answer lies in that based on the Darwin evolutionary theory. What happens here? It tells us the survival of the fittest. Those who survive in the adverse condition will survive and they cell and they maintain their cell structure and the ATP generation will be happening and growth. So whoever fit to the particular environment, they will survive and others will obviously die. So this is how the metabolism concept has evolved. And if you see the metabolism, if there is any change in the metabolism or if there is any change in their behavior of catabolism or anabolism or some kind of proteins that are not appropriately synthesized during this process, for example, the glucose is converting into pyruvate through the glycolysis cycle, right? And this pyruvate is converting into acetate through TCA cycle, where the ATP is generated through various cycles. And in order to catalyze these reactions, there are numerous proteins that are involved during the process. If any one of the if any one of the proteins they do not function at they do not perform its function properly. For example, any kind of mutation or changes in the protein structure that will leads to reduced number of ATP in the whole metabolism cycle. And this we call it as the deficiency of metabolism or or we call this condition as metabolism deficiency. And this is how the metabolism deficiency occurs if there is any deficiency in the protein that are not able to produce appropriate amount of ATP for their cell survival or cell growth or cell maintenance. And this is where the syndrome happens. As one of the audience asked about the metabolic syndrome, it is nothing but there is a deficiency in the metabolic risk factors, right? So what happens here due to in deficiency of a particular protein, as we have seen, in order to convert the sugar into pyruvate, or glucose into pyruvate, there are numerous proteins that are involved. And this deficiency in any one of the protein will cause a deficiency in the whole cycle, which reduces the ATP generation. And if we see in terms of metabolic syndrome, there will be any deficiency. It can be the deficiency in a particular protein can cause, if you say in humans, it can cause an high blood pressure or there will be higher cholesterol levels. So any deficiency in a particular protein will cause us the metabolic syndrome. And we call this condition, and the metabolic syndrome is not a single disease. What happens here? A combination of related condition will be happening. So what happens? The risk of the survival or the risk of disease is higher, and there could be a numerous factor. For example, a single disease can cause an high blood pressure or high blood sugar level or a higher triglycerides, or it can lead to higher cholesterol level. So this syndrome is not a single disease. It is caused by multiple epigenetic factors. And what environment does the human have? I have given an human example. You can correlate with the microorganism where a particular microorganism survival condition. For example, the high temperature microorganism cannot survive in the cold temperature. So we will see a numerous changes in their metabolism, their maintenance level, and their ATP generation. And this is important for understanding the evolutionary of ecology. So moving back to the concept. So the survival of the fittest, it not only applies to the ancient and modern microorganism, it not only applies to the ancient, it also applies still now to the modern microorganism under all the environmental conditions. And if you see the key types of physiological and ecological pressure that confront the microorganism in nature, and how do we call an ecology is successful and failure. So if one can say an ecology or the environment is successful when if there is a nutrient rich environment where an abundance of carbon and energy content is higher, and if there is a nutrient rich environment, there is an higher growth, survival, and the population expansion also. But what happens if the ecology is failed? What do we observe? There will be an adverse condition, like you can observe a population decline or death in the population of cells or elimination of the particular population from that habitat. And when it happens only in the adverse condition where we could see there is a starvation or competition between the two microorganisms, or there is an higher UV light or oxidative stress, which interferes with the population survival and growth. So from this, we could understand that the microbes also faces a lot of challenges. One, but the one that survives and adapts, they fit into the environmental condition. Those that can't fade const. So what happens here? There is a ecological success and failure. There is a balance between the one population growth over the other population, and this ecological pressure drives the microbial diversity and the evolution. And this is how we say the an environment is success when they have an increased population size and abundant growth is observed. And whenever it fails, we call it as ecological failure. And if you see the growth condition, especially the microorganism undergoes two growth strategy. One is R selected species and K selected species. And how do we segregate them? How do we segregate an ecosystem into R selected and K selected species? We will see how do we segregate them is that Whenever there is a higher growth or higher rate of reproduction, or the reproduction occurs very quickly, they take advantage of every bit of nutrients and the space availability in an uncrowded habitat. We call these kind of species as or selected species. For example, E. coli. When E. coli is placed in a nutrient rich environment, what do they? They will undergo a rapid rate of reproduction, right? So, in an uncrowded habitat, the E. coli will be growing very higher rate of growth. So, we can classify them as or selected species. What about the case selected species? It is not here. What happens? They doesn't grow overnight. Their growth is very slow, conserve its energy, and they survive for decades. So we call these kind of species and classify them in the case selected species. So in short, we can say that the or selected is nothing but their growth will be quick and explosive and short-term success. Whereas we can compare the case selected as they will their growth is very slow but steady, and they will be going for long-term survival. So these are all the growth strategy, and we will place the appropriate species in the RN case selected species. So we will move on to the concepts. The next concept is thermodynamics. Why we need to study the thermodynamics in the microbial environmental biotechnology aspect? Because the first is we have to understand the flow of electrons or flow of energy, because energy drives the metabolism, right? So you have to understand the flow of energy, and the thermodynamics is nothing but the science of studying these kind of energy flow. It decides whether the reaction, a particular reaction, needs to be happen or not. So what happens here is it tells us that the particular reaction which is energetically favorable or not. So it tells us which reaction needs to be carried out, whether it is possible or not. And there are different types of reactions are also there, right from dissolution, precipitation, acid base reactions, sorption, desorption reaction, and oxidation reduction reactions. So these are all kind of reactions that normally observed in the thermodynamics. And with respect to the environmental biotechnology aspect, with respect to the environmental biotechnology aspect, we will be more focusing on this oxidation and reduction reaction. And these oxidation reaction, reduction reaction, we have already seen in the previous sections also how microbes loses their electrons and gain the electrons in order to catalyze or oxidize the organic substance. And and if you see, the most important term in the thermodynamics is their del G value. What is del G? Del G is nothing but the free energy, the energy required to catalyze some kind of reaction, that is converting a reactant into a product. For example, if you see in the graph, the reactant A plus B will be converting into product C plus D. So in order to catalyze this substance A plus B into C plus D, the system needs to spend some kind of energy. And this difference, we call it as the free energy. And what happens if this value is negative? If del G is negative, that means the reaction is very spontaneous. That is, it is very quick enough to carry out this particular type of reaction. And if the del G value comes to be positive, then it is a non-spontaneous reaction. What does it mean? It means it the system needs high input of energy in order to convert the reactant into the product. So what does this del G tells us? It tells us the spontaneity of the process at that particular temperature and pressure. So here we will first try to identify the resources that are exploited by the microorganism. Resources is nothing but the carbon and energy source and how it facilitate the ATP generation. And it is the thermodynamically unstable resource that have provided the selective pressure for microbial ecology production strategy through the evolution. And we will see with the examples. For example, as we have seen, one of the most reactions that microbes drive is oxidation reduction reaction. And if you see the chemisynthetic organotrophic life, what does it means? It is nothing but it will use organic compound as the substrate, that is carbon source as well as the energy source. Here what happens? The microbes takes the organic material, they mix with the oxygen and the oxidizable compounds and it sets the stage for metabolism. And what happens? It fuels their metabolism and keep the ecosystem viable. And here the important point to note that here the organic compound act as in both carbon and energy source. Whereas if you see the metabolism of chemosynthetic lithotrophs, the microbes takes the inorganic materials like H2, NH3 ammonia and H2S with oxygen and other oxidizable material, which sets the stage for chemisynthetic lithotroph life. So depending upon their resource, they will set the stage for their metabolism. So if you see here, the water plus carbon dioxide, if it uses light as an energy source, we call it as a phototrophs. And it what happens? It will reduce the carbon and they will use for metabolism through various oxidation and reduction reaction. And if you want to understand the oxidation reaction of a particular ecosystem, you need to understand the concept of electron acceptor. And we will see the concept of E naught and PE later. But as of now, you can understand that the most energetically favorable reactions are first is O2 reduction. What happens here? The oxygen will be acting as an electron acceptor and it will be converting into water. So this is the more favorable reaction because it has an higher E naught value and higher PE value. And we will be defining these terms in the upcoming slides, followed by the oxygen reduction. The next energetically favorable reactions are denitrification, where the nitrite and nitrates will be converting into nitrogen gas, followed by MnO2, followed by MnO2 reduction, followed by NO3. So this is the hierarchy of energetically favorable reaction to the unfavorable reaction. And the least unfavorable reaction is the methane termination or hydrogen production. What happens here? The methane is produced from the CO2. So this is the least favorable reaction because it has the lower E naught and PE value. And as I said, what do you mean by PE? PE is nothing but it represents the intensity of the electron activity in a given ecosystem. And how do we say pH? pH is nothing but the number of proton that are present in a given system. That is the H plus ions that are in the given system. We call it as pH. If there are very less number of pH plus in the given in the given environment, what do we call it as? If the H plus protons are less, what do we call it as? It is acidic or basic. If there are less number of H plus ions. If there are very less amount of H plus ion, we call it as acidic. If there are higher number of protons, we call it as basic, right? So similar to that, the PE tells us the intensity of the electron activity. And how do we measure it? How do we calculate it? It is nothing but the negative logarithm of the electron activity. And it tells us the tendency of a solution. to whether it is accepting an electron or donating an electron. So if suppose a particular solution has a lower p-value, what does it tells? It tells us that higher electron activity is observed. So the solution favors the reduction reaction. Whereas conversely, an higher PE indicates a low electron activity and it always favors the oxidation reaction. And this is more important concept and you need to understand this term in order to understand the oxidation reduction mechanism. And if the value is lower, it will always favors the reduction reaction. And if the value is higher, it will favors the oxidation reaction. And the next term that you need to understand is the E naught, which is nothing but the standard electrode potential. What does it tells us? It measures the chemical species that undergo reduction reaction under standard condition. So what happens if the E naught is positive? If E naught is positive, then the solution has higher tendency to go for an reduction reaction. That is a strong oxidizing agent. It acts as a strong oxidizing agent. Whereas if the E naught is negative, it has a greater tendency for the oxidation reaction. So these two important terms determines the system whether the energy should be favorable or unfavorable. And these are all the 8 different processes that normally observed in the carbon rich habitat. The first is the aerobic respiration. What happens? What do you mean by aerobic respiration? Anyone? Aerobic respiration refers to or aerobic process. What do you mean by aerobic process? Yes, Ankita, you are right. Yes, to him, aerobic process means a process occurs in the presence of oxygen. So it will use the oxygen as an electron acceptor and convert into water. And if you observe the PE value, it is positive, and more importantly, if you observe the del G, it is highly negative, minus 125. And in denitrification, what happens? The nitrates is converting into nitrogen. And in manganese reduction, MnO2 is converting into MnCO3. That is, the manganese state is reduced to Mn3 to Mn3 plus to Mn2 plus. There are likewise there are 8 reactions, and the least favorable reactions are methanogens. What happens? The carbon dioxide is converting into methane, and the carbon dioxide is converting into acetate in the case of acetogenesis. And in all this process, if you observe the del G value, as we have understood the term del G value, what does it implies? It tells us the spontaneity of the process. And anyone can tell what happens if the del G is negative? If del G is negative, it favors spontaneous process or non-spontaneous process. If del G is negative, if del G is negative, the reaction is more spontaneous. That is, they are more energetically favorable. There is a higher chance of reaction that occurs. For example, if you observe in the last row, the del G is very high. That is, they are negative, minus 125 for aerobic respiration. So what happens in a given ecosystem? If there is a presence of oxygen, it always takes oxygen as the first priority. For example, we will consider two situations where oxygen is present and carbon dioxide is present. So from the table, we could understand that the del G value of oxygen for catalyzing aerobic respiration is minus 125 and for methanogenesis is minus 23. So as we have seen that if del G is negative, they are more spontaneous. So the environment favors oxygen as the more favorable electron acceptor than the carbon dioxide. If suppose if the environmental contain nitrate and carbon dioxide, still it prefers the nitrate as the electron acceptor because their del G is very negative when compared to the methanogenesis. And this is how the reactions are favored and the process are occurred. And we will see examples, numerous examples regarding how in an environment this oxidation reduction occurs. So the first example we will see a laboratory demonstration where the sequence of thermodynamically predicted process that generates the PMF, which is proton motive force, and hence ATP is formed. So for this experiment, we will first take an closed lid where we will be adding 10 grams of soil and we will be adding water and we will be adding organic substrates like CH2O, that is we will be providing carbohydrates, that is one of the substrate, and we will be providing all the electron acceptors like O2, NO3, SO4, Fe2 and CO2. So we will be providing all the oxidizable electron acceptors and we will be providing the substrate and the soil. And we know that the soil will contain numerous millions of microorganisms, right? And we will test what is these microorganism is assimilating first. And why do we perform this experiment? What is the rationale behind it? We will be providing carbon and energy for the heterotrophs, and organic substrate is the electron donor, and oxygen and other electron acceptors are also provided. And what do we measure? With respect to the time, we will be observing the changes in the ecosystem. And what happens? We will be removing the water and we will be analyzing the presence of thermodynamically patterns will be observed. And what happens? From the graph, we could observe that the most energetically favorable reaction occur first and the least at the last. And from the graph, we could say that oxygen is first consumed by the aerobic heterotrophs, followed by the denitrifier bacteria, followed by the Mn reducer bacteria, Fe reducing bacteria, SO4 reducing bacteria, and at last the archaebacteria. And how do we found out this? By observing the products that are formed during the reaction. If you observe in the first graph, if it is assimilated the oxygen as an electron acceptor, see the amount of water is increasing at the first, followed by ammonia, Mn2 plus, Fe2 plus, H2 plus, and CH4 minus. This tells us that aerobic process is preferred more because they have del G value highly negative when compared to the CO2 del G value. That is why they are preferring O2 as the first option for catalyzing the substrate. So this is one demonstration at the lab scale level. And if you move on to the field level, for example, we will take one example which is oil spill. What happens in the oil spill? If you observe the environment of the oil spill, you will observe the anaerobic condition near to the oil spill, followed by the aerobic condition at the farther from it. That is why in the graph, in the picture, we could observe that low oxygen to the high oxygen. What does it means that here near to the oil spill, there will be very less amount of oxygen. As we move away from the oil spill, there will be a higher amount of oxygen. As the situation is clearly stated, now we could observe what kind of reaction could have taken place. What kind of reaction could have taken place as there is very less amount of oxygen is present. This is similar to that of anaerobic condition, right? So methane will be the first product that is formed by assimilating what? by assimilating carbon dioxide by the methanogenic archaea, followed by H2S formation, followed by Fe2, Mn2. At last, it will utilize oxygen as a substrate and form the water as the byproduct. So this is how the microorganism drives the substrate and helps in cleaning the environment. This is in case of aerobic environment where oxygen is preferred first, whereas in the anaerobic environment, CH4 will be preferred first. So this is the two different situation that we encounter for the electron accepting process. As we have understood the electron acceptor process, the next key question that you need to ask is the how the electrons are transferred. I hope my voice and PPT is visible. Whether my PPT is visible? Thank you, Tuhin. Okay, we will continue the logic of electron transport, where I was explaining why electron transport is important. Why? Because the movement of electron drives the microbial cell. What happens here? It helps in generating the ATP. And why the ATP is very important? It helps in fueling the cell maintenance and the cell growth. Without ATP, what happens? The life stops and the cells do not function it normally. And this is the ATP is regarded as the energy currency of the cell. And if you see the cellular metabolism, as we have seen earlier, it is classified into catabolism and anabolism. What do you mean by catabolism? Catabolism is nothing but breaking down the substrate for the ATP generation. Whereas anabolism tells us using the fuel of ATP, they will generate new molecular buildings like low molecular weight organic compounds into new cells, or they provide maintenance for the existing cells. And if you see the catabolism and anabolism, what happens? The substrate is converting into a ATP, where oxidation of reduced substrate will be happening. How it is happening? It will happen through substrate level phosphorylation followed by oxidative and electron transport chain. Here, the dissimilarity reduction of O2, NO3, FeOH thrice will be happening. Whereas it's quite opposite of catabolism. What happens here? The ATP will be used in order to generate a new cell materials or biomass. So here biosynthesis of new cells will be occurring by creating a strong C-C bonds. And here assimilatory reduction of CO2, NO3 or SO4 occurs. So this is the basic the differences between the catabolism and anabolism, where the substrate get converted into ATP in the case of catabolism, whereas in anabolism using the ATP new cells are formed. Like this is the destructive process, this is the constructive process. And if you see the catabolism process, where breaking down of glucose will be happening. So we will explain this process first. What happens here? First, glucose will be converted into pyruvate through glycolysis cycle, and pyruvate will be converting into aceto-CoA, and this will be entering into the TCA cycle, which is tricarboxylic acid cycle. What happens here? Once it has completed its cycle, it will generate numerous NADH, FADH, and ATP. This this NADH or FADH needs to be converted into some kind of energy, right? So what happens? This will be moving into the electron transport chain where ATP will be formed through ATP synthase mechanism, where oxygen will be used as an electron acceptor and it will be converting into water. So by this mechanism, you could see a numerous ATPs are formed at the each stage, and we could see some FADH2, NADH2 is also formed. Then how much, literally from one glucose, how much ATP is formed? We could answer this question by, if you see in the glycolysis cycle, 2 ATP is formed along with 2 NADH. For per NADH, you will be generating 3 ATP. So here we have 2 NADH. Then 2 into 3, 6 ATP is formed. So at the end of the glycolysis cycle, you will be having 8 ATP molecule. Whereas in the case of citric acid cycle, you will be getting 1 ATP, 4 NADH and 1 FADH. With 1 NADH, we already seen 3 ATP will be formed. So it is 4 into 3, which gives the 12 ATP. And from 1 FADH, you will be getting 2 ATP. Right? So if you calculate this, 1 plus 12 plus 2, you will be getting 15 ATP. Then how 30 ATP came? Because by through glycolysis cycle we are getting 2 pyruvate. With 1 pyruvate we will be getting 15 ATP. If it is undergoes another cycle of TCA cycle, it will generate another 15 ATP. That is why we are getting 30 ATP. So totally how much? 8 ATP from the glycolysis cycle and 30 ATP from the citric acid cycle. So eventually we will be getting 38 ATP molecules from oxidizing a single glucose, which is 6 carbon cycle, 6 carbon compound. And this is how the whole catabolism event occurs in the glucose molecule. And in order to tell in depth, this microorganism will carry out the catabolism by using substrate, oxidizable substrate, to deliver the electrons to the electron transport chain. What happens here? Through glycolysis TCA cycle, it will produce higher ATP. It will produce higher amount of ATP. It will produce higher amount of ATP. What happens in the absence of O2? Any idea what happens in the absence of O2? In the absence of O2, they will undergo anaerobic mode of respiration. So instead of oxygen as an electron transporter, they will use other electron transporters like nitrates, sulfates, and CO2. So the free energy gain under such situation follows the thermodynamic hierarchy. What thermodynamic hierarchy? First is oxygen, followed by nitrate, followed by Fe2, Mn2, followed by Fe2, and at last CO2. So this is the hierarchy that we have seen in the previous slides. And the last important concepts that we are going to see in this session before moving on to the MCQ is the syntropy concept. So what is syntropy? Syntropy is nothing but two different microorganisms, they cooperate each other in order to degrade a particular substance. Why they are cooperating each other? Because if they act alone, they cannot degrade the particular compound. We will be seeing examples, numerous examples, but to understand this, it is nothing but two different microorganisms will cooperate each other in order to degrade the substance. So without syntrophy, many of this pathway don't work. And the syntrophy association of microorganisms, they are often critical for the carbon-driven ion reduction, manganese reduction, sulfate reduction, acetogenesis, and methanogenesis. And what happens here? They are most of these reactions are from secondary fermentation. What do you mean by secondary fermentation? The microbes, they don't ferment the original substrate. They ferment the waste products that are generated from the primary fermentation. And we will be seeing the flow of carbon and electrons in the case of anaerobic food chains. So if you see the anaerobic condition, first the organic compound, which is high molecular weight polymers, if you can take any kind of waste, first what happens, it will undergo the primary fermentation where we will be uh, getting fatty acids, alcohols, lactate, H2 and O2. So iron or manganese reducing bacteria will use these fatty acid, alcohols and lactate in order to reduce the secondary metabolites, which is acetate and H2. So at last what happens, the intermediate product with help of the methanogenic archaea, they will convert at last into CH4. So from this we could understand it is not one organism, it is two or more organisms. In this case, F and Mn reducing bacteria first cooperating each other in order to form the CO2, and with the help of these reducing bacteria, they will be converting the CO2 and converting into CH4, which is helped by the methanogenic archaea. And we will see in the syntrophy level why H2 is very important, because H2 acts as a currency. Microbes used to trade each other. One microbe produces H2 which will be consumed by the other. This phenomena we call it as syntrophy, right? So by keeping the balance in the ecosystem. So we will take one example and we will clarify the doubts why H2 is very important. First, we will take the ethanol. When ethanol combines with the water, the microorganism which is Paletobacterium will produce acetate, along with it will produce hydrogen and H plus protons. So if you see this reaction, it is del G is positive. What happens if the del G is positive? If del G is positive, it is spontaneous or non-spontaneous reaction. If del G is positive, it will be a non-spontaneous reaction. That is, it needs to spend higher amount of energy. This kind of reaction is often unfavorable for the particular environment. That is why the concept of syntrophy is evolved. What happens? This hydrogen will be utilized by the other group of microorganisms, which is methanogens, that will help in converting them into methane, a valuable product. And if you observe the del G naught value, it is highly negative. What does it tell? It makes the reaction energetically favorable. So by this we understood the concept of syntropy, where two different type of bacteria, one is electron and methanogen, helps in converting the ethanol into methane by maintaining the overall reaction as favorable reaction. And one more example, we can also see that the cellulose undergo primary fermentation where organic acids, alcohols, and H2 is formed. And if you see in the first step alone, the del G value is positive, which is not energetically favorable. But once this organic acids, alcohols, or H2 is consumed by different group of microorganism, the overall del G is becoming negative. So in order to maintain the overall reaction negative, two or more different microorganisms, they cooperate each other and they try to make balance and make the reaction energetically favorable. And this is how the concept of syntrophy evolves in the microbial ecosystem. So this is all for the, and this is for all the to gain only 2 ATP through glycolysis. No, along with 2 ATP, there will be a production of NADH and FADH. And as we could see that there will be 8 ATP which will be formed at the end of the glycolysis cycle. So, so this is all for the today's class. Can we move on to the MCQ? Can we move on to MCQ now? So, okay, okay. So can we move on to MCQ? So this should be an interactive session. So I will be first asking the question. You can put your answers in the chat box, or you can answer by unmuting the mic. Anything is fine for me, but it should be interactive so that it will be okay. Okay. So I think we can move on to the MCQ. So for this session, these are all the sample MCQ. The first question is that mushrooms are option A, photosynthetic part of underground multicellular fungi. Option B, fruiting body of underground fungi. Option C, mutualistic association of algae and fungi. Option D, aggregates of protists. Option E, aggregates of elements. So among this, which suits for mushrooms? Mushrooms are, you can post your answers in the chat box, or you can unmute your mic and you can answer the question. Mushrooms are option B. Okay, okay, Ankita. Okay, Tuhin. Any other answer? Mushrooms are okay. Option B. Okay. So let's see the answer. Yes, it is option B. Mushrooms are fruiting body of larger fungal mycelium that grows beneath the ground. So why not other options? So first is photosynthetic part of underground multicellular fungi. Yes, it is a fungi, but fungi do not perform photosynthesis, right? What do they obtain the energy from the organic matter. So this option A is wrong. If you see mutualistic association of algae and fungi, what do we call the mutualistic association of algae and fungi? We call it as lichen. It is not mushrooms. Mushrooms are solely fungi. Option D, it is not an protist, it is not an almanthus. So it is no way connected to the fungi. So it is mushrooms are solely fungi. Yes, the answer is option B, fruiting bodies of underground fungi. So we will move on to the next question. Which of the following organisms are beneficial for wastewater treatment? Option A, helminths. Option B, protozoa. Option C, fungi. Option D, cyst. Option E, amoeba. So which one of the following are beneficial for the wastewater treatment? Your answers are option B, okay, option C and B. Okay, option C and option B. Okay. Any other answer? Okay, now we will see the answer. It is option C, fungi. And if you have gone through the lecture 18, professor has emphasized the importance of fungi in the wastewater treatment. And where we have studied that fungi are important degraders of polymers, and they are used in the composting and biodegradation of toxic organic substance. And fungi are used in environmental engineering application, where in composting, soil bioremediation, and biodegradation of xenobiotics. And this is why fungi are especially used in the wastewater treatment. Okay, we will move on to the next question. Which of the following is not an essential nutrient for the microorganism? I will repeat the question. Which of the following is not an essential micronutrient for the microorganism? Option A, tungsten, which is W. Option B, vanadium, V. Option C, selenium, Se. Option D, arsenic, As. Option E, copper, Cu. So among this, which is not an essential micronutrient for the microorganism? And your answers are, okay, it is D. D. Any other answer? Option D, arsenic. Okay. Any other answer? Okay. Now we will see the answer. Yes, it is option D. Arsenic is not an essential micronutrient as it is toxic to the most of the microorganism. So it doesn't comes under micronutrients. And what do we call a micronutrient? Anyone? Micronutrient is what is micronutrient? Micronutrients. Yes, yes, Steven. Yes, Ankita, it is correct answer. So these micronutrients they require in the very minute quantity for their functioning. So if you see the first option, tungsten, especially for anaerobic or thermophilic bacteria, it acts as an cofactor. So it is one of the essential micronutrient for anaerobic and thermophilic bacteria. And if you see the vanadium, especially for nitrogen fixing bacteria, it is one of the essential micronutrients that is observed. And selenium, it is its form selenocysteine. It acts as for redox reaction and other metabolic process. And if you see copper, copper acts as an cofactor for many of the redox active enzymes. So it is also one of the essential micronutrient. So if you see the tungsten, vanadium, selenium, and copper are essential micronutrients for different microorganism, whereas arsenic is toxic. So we will move on to the next question. The next question is, which of the following statement is true? You have to identify among this statement which is true. Option A, proteins are most abundant macromolecule in any cell. Option B, the glycogen is used for storage only by the eukaryotes. Option C, all components of the complex medium are precisely quantified components. Option D, defined medium can show batch to batch variation. And option E, the storage compound PHB can be formed during the oxygen starvation. So among this statement, which is true? You can post your answers. I'll repeat the question. Which of the following statement is true? Okay, option A. Proteins are most abundant macromolecule in any cells. Any other answer? Ankita? Any other answer? Tuin and Ashwini said option A. Okay, okay, Ankita, we will see the answer. It's okay. So the answer is option E. Storage compound PHP can be formed during oxygen starvation. We will see the solution first. So if you have gone through the lecture 19 where Professor has discussed on the storage compound, where polyhydroxybutyrate accumulates under excess carbon source as well as the oxygen limitation, and it serves as the carbon storage reservoir. And that is why the option E is true. Storage compound PHP can be formed during the oxygen starvation. So it will be formed during the oxygen limitation. And we will see now the other options why it is not correct. The first option, which is proteins are most abundant macromolecule in any cells. So yeah, yes, it is partially correct because protein is one of the abundant macromolecule. What are the other abundant molecule in any cells? RNA, DNA is also often considered as macromolecule, carbohydrates, fats. Sorry, RNA and DNA is also considered as an abundant macromolecules. Phospholipids, yes, yes, it's correct. So protein is one of the most abundant, not the only most abundant. And the option B, the glycogen is used for storage only by the eukaryotes. No, it is not correct. It is not only for eukaryotes, it is also applicable for the prokaryotes as well. All components of the complex media are precisely quantified components. This is obviously false because complex media means their media components are unknown or it is variable concentration. So it is not precisely quantified. It is approximate. It is unknown compound. So this is also false. And defined media can show batch to batch variation. And what do you mean by defined media? Anyone? Defined media? In defined media, whether the nutritional components are exactly known or we don't have exact nutritional components. In defined media, their nutritional components are well known. So if the media components are well known and their concentration are well defined, then we can observe batch to batch variation is very, very less or very minimal. So because of their precise composition. That's why option A, B, C, D are false and option B, E is true. So we will move on to the next question. Next question is, which of the following statement correctly describes the cell cycle stage? Option A, G1 begins after DNA replication is completed. Option B, M phase is the period between G1 and G2. Option C, the S phase is the period of DNA replication. And option D, G2 is followed by S phase. Option E, G0 is the period between completion of cell division and initiation of DNA replication. So among this statement, which is true regarding the cell cycle stage? You can post your answers. I will repeat the question. Which of the following statement correctly describes the respective cell cycle stage? Option C. Okay. Option D also. Okay. Any other answer? Now we will see the answer. Yes, the answer is C. S phase is the period for DNA replication. So if you see the cell cycle, you can observe that in S phase, DNA synthesis is happens. Why not the other answers? G1 begins after the DNA replication is completed. This is false, right? G1 begins before the DNA replication is completed. So option A is false. And option B, M phase is the period between G1 and G2. If you see the cell cycle, Between the G1 and G2, S phase is there, not the M phase. So M phase is not the period. S phase is the period between G1 and G2. And obviously S phase is the period of DNA replication. And why D is not true? G2 is followed by S phase. No, it is false. S phase is followed by G2. So if you see the cycle, S phase is followed by G2. And last one, G naught is the period between completion of cell division and initiation of DNA replication. This is also false because G naught is the resting phase. That cell enters from the G1 phase. Here what happens, neither they are actively dividing or prepare its cell to divide. So that's why option A, B, D and E are false. And the answer is C. So we will move on to the next question. As we have seen in the earlier lectures, if you have attended, you could answer this question. So the next question is metabolism, replication and dash or critical triads of life. Senescence, persistence, quiescence, heredity and mutation. Among this option, which is correct, which is considered as one of the critical triads of life. I will repeat the question, metabolism, replication and dash or critical triads of life. It's okay, Twin. So now you can answer the next question, metabolism, replication and dash or critical triads of life. Option D, okay, heredity. Any other answer? Any other answer? Option D, okay, option D. Any other answer? Any other answer? Metabolism, replication and which one is critical triads of life? Okay, we will see the answer. Yes, it is correct because all living beings must perform metabolic reaction. They have to self-replicate and pass on their genes. And what do you mean by heredity? Heredity? Anyone? Heredity means here heredity refers to heredity is nothing but passing the information or trait from one generation to the another generation. Right. So why not the other what about the other terms? Senescence refers to the process of aging. So this is not one of the fundamental triads of life. Persistence refers to surviving in the harsh or adverse condition. Yes, it is important for survival, but it is not one of the critical triads of life. Quiescence refers to being in the state of dormancy, that is, in the hibernation condition. And mutation refers to change in the base pair. So, metabolism, replication, and heredity are the basic form of life. So, we will move on to the next question. Next question is, which one of the following statement is true? Option A, only light can be used as an energy source. Option B, photosynthetic organism, they do not need a carbon source. Option C, same substance may serve as an energy and carbon source. Option D, energy source is always utilized to produce precursor molecule. Option E, ATP is produced only from the chemical source. So among these statements, which is true? You can post your answers. Option C, okay. Same substance may serve as energy and carbon source. So any other answer? Any other answer? You can take your time. So which one of the following statement is true? Option A, only light can be used as an energy source. Option B, photosynthetic organisms do not need a carbon source. Option C, the same substance may serve as an energy and carbon source. Option D, energy source is always utilized to produce precursor molecule. Option E, ATP is produced only from the chemical source. Option C, okay. Okay, let's see the answer. Yes, it is option C because some substance like glucose, it can act as an energy and carbon source. Now we will see why not other options. So only light can be used as an energy source. No, not only light. Chemical energy is also used as an energy source. And what do we call? What do we call if it uses light as an energy source? What do we call if it uses light as an energy source? What do we call that microorganism? Light, it is lithotrophs. What if it uses chemical as an energy source? It is chemotrophs. So, not only light, it can also use the chemical source in order to for energy sources. The second option, photosynthetic organisms, they do not need a carbon source. No, it is false because it requires CO2 as a carbon source in order to build new nuclear molecule. And option D, energy source is always utilized to produce precursor molecule. No, they don't directly produce them. ATP is produced only from the chemical source. It is wrong, right? Light is also used to generate the ATP and we call them as the phototrophs. So the option C, the same substance may serve as an energy and carbon source. That is, glucose can serve as both carbon and energy source that we have seen in the earlier lecture, that is through glycolysis, TCA and electron transport chain. The glucose is not only serve as an carbon source, it acts as an energy source for ATP production. So we will move on to the next question. Which of this following nutritional type best describes for the human metabolism? For human metabolism, which of these nutritional type will suit? Option A, chemisynthetic organotroph. Option B, chemisynthetic lithoheterotrophs. Option C, photosynthetic heterotrophs. Option D, chemisynthetic lithoautotroph. Option E, photosynthetic autotrophs. So among these options, which suits best for human metabolism? You can type your answer. It's okay, Ankita. It's not a problem. So what is the answer for this? Which of this nutritional type best describes the human metabolism? Okay, option A, chemisynthetic organotrophs. Okay. Any other answer? Okay, option A. So what do you mean by chemisynthetic organotroph? Here, what is the carbon and energy source? Anyone? Chemisynthetic organotroph. Okay, glucose. Yes, it is correct. Option A, human utilizes glucose as the carbon and nitrogen source. What happens here? Here, organic compound acts as both carbon and energy source. So we will see the rest options. Chemisynthetic lithoheterotrophs. What happens here? Here the energy is inorganic compound. So humans don't use the inorganic compound as the primary energy source. So option B is wrong. Next option is photosynthetic heterotrophs. So plants only perform photosynthesis, right? Humans do not perform photosynthesis. So obviously option C and E is eliminated because option C and E perform photosynthesis. While human metabolism, they do not perform photosynthesis. Option D, chemisynthetic lithoautotrophs. What happens here? Here also it uses inorganic compound as an energy source in order to produce organic compound. So humans are heterotrophs, not the lithoautotrophs. So option D is also false. So the correct option is option A, chemisynthetic organotroph. So we will move on to the next question. So which one of the following process directly result in ATP formation via membrane-bound ATP synthase? I will repeat the question. Which one of the following process directly result in ATP formation via membrane-bound ATP synthase? Option A, oxidation of reduced electron carrier. Option B, reduction of oxidized electron carrier. Option C, translocation of H plus out of the cytoplasm. Option D, translocation of H plus into the cytoplasm. Option E, ATP hydrolysis. So I will repeat the question. Which one of the following process directly results in ATP formation via membrane-bound ATP synthase? You can post your answers. Option D. Okay. Any other answer? Option D. Okay. Any other answer? Option D. Any other answer? So let's see the answer. Yes, it is correct. Option D. Translocation of H plus into the cytoplasm. So what happens? How the generally the H plus will be transferring? It will be transferring from the extracellular or transmembrane region to the cytoplasm, which powers the ATP machinery. Why not the other options? First is oxidation of reduced electron acceptor. What happens? It is process that occurs in the electron transport chain. This needed for the ATP synthesis, but this is not directly or via through membrane-bound ATP synthase. Whereas the reduction of oxidizing electron carrier, it is a reverse step of electron transport chain. If it is a reverse step of electron transport chain, there will be no ATP synthesis occurs. So option B is wrong. And option C, translocation of H plus out of the cytoplasm. No, as we have already seen, H plus translocation into the cytoplasm. Then option E, ATP hydrolysis, and the term itself suggesting at that here the breaking down of ATP occurs, not the formation of ATP. So here ATP is broken down and releases energy, so it does not generate the ATP. So the correct answer is option D, translocation of H plus into the cytoplasm. So we will move on to the last MCQ, which is which one among the following is most energetically favorable terminal electron acceptor. So we have seen a series of electron acceptor, right? And we have seen which is most energetically favorable based on the del G value. So among this one, which is most energetically favorable? Option A, NO3, option B, Mn4 plus, option C, Fe2 plus, option D, H2, and option C, CO2. So you can post your answers now. Option A, okay. NO3, okay. Option A, okay. Option B, Mn4 plus, okay. So before moving on to the answer, we have seen the del G, right? Del G represent what? Del G means del G stands for del G stands for yes, it gives free energy. And what happens if the del G is negative and what happens if del G is positive? First, we will see if del G is negative. If del G is negative, what kind of reaction? Yes, it is correct, Ankita. Twin, it is spontaneous. And if it is positive, it is non-spontaneous. So, if we observe the question for an energetically favorable reaction, whether del G should be positive or negative for a energetically favorable reaction. Del G should be negative. Yes, it is correct. And we have seen the order of electron acceptors also. Okay. So now we will see the answer. Yes, it is correct. A, NO3. So if you say in terms of energy yield, nitrate is considered as the most beneficial electron acceptor for many microorganisms under anaerobic condition, allowing them to generate significant amount of ATP through its reduction process. So if you see Mn4 plus, it is also one of the electron acceptor which will be converting into Mn2 plus, but it is not energetically as favorable as the NO3. Whereas Fe2 plus, it is also one of the electron acceptor, but their reduction is less energetically advantageous when compared to NO3. Whereas H2, it is not an electron acceptor, it is an electron donor. It is oxidized in order to generate energy. And option E, CO2, it is a final product of metabolism. It is not an electron acceptor. So the correct answer is NO3. So I will ask one question. If you have NO3 and O2, then in this case, which is energetically favorable reaction, whether it is NO3 or O2? Okay, to win O2. Any other answer? Yes, it is correct, to win. It is O2 is more energetically favorable because what could be the reason? Why O2 is more preferred than the NO3? Because as we have seen in the table, right, 8 reactions that are common. If you see the aerobic mode of respiration, their del G is highly negative when compared to the NO3. If you see the del G value, I will also show. if you see this process, aerobic respiration, where the del G is minus 125. Whereas if you see the denitrification process where NO3 acts as an electron acceptor, their del G value is only 119. When you compare the del G between these two, the del G value is more negative in case of aerobic respiration. That is why it is more energetically favorable reaction. You are correct, Tuyin. So, so these are all the sample FCQs that I want to discuss for this week. So you can ask your friends also to join in this session because these sample MCQ will prepare you for the final exam as well. So try to attend as much as class possible. So in this class we have seen many important concepts like centropy concept, logic of electrons, the effect of carbon and energy source in catabolism and anabolism reactions. So these are all the concepts that we have covered, and also we have seen samples MCQs regarding this. And thank you for patiently listening to this lecture. Try to attend as much as class possible so that you can prepare yourself for the final exam by solving this sample MCQs. And if you have still any doubts, you can mail me or you can post your queries on the NPTEL discussion forum. Our team will address those queries as well. And if you miss my any of the earlier lecture, you can visit my YouTube channel Ajit Kumar underscore B, or you can visit the NPTEL Environmental Biotechnology course where in the problem solving session, the recording sheet is there. You can access all the PPTs as well as the recorded video sessions. And still, do you have any doubts? If you have doubts, you can mail me also. Do you have any doubts? Okay, Ashwini. I hope you have all submitted the assignments. And if you have any queries regarding the assignments, you can post your queries in the discussion forum as well. And if you have missed out any of the earlier videos, you can revisit in the recording sheet. So any doubts? Okay. So can we wind up the session? Thank you, Tuhin, Ankita, Ashwini. Thank you for patiently listening to this lecture and do not miss the future lectures as well. There will be numerous MCQs that we will be solving. Okay, thank you all. You may leave the session now. Thank you all.